Bismillahirrahmanirrahim. Assalamu alaikum viewers. Today we are going to discuss glycolysis. Glycolysis is actually the first step of cellular respiration, which is a combination of two words, that is glyco, which means carbohydrates, and lysis means breaking. So glycolysis is the breakdown of glucose molecule into two pyruvate molecules. Glucose is a 6-carbon sugar which is broken down into two molecules of pyruvate at the end of glycolysis. The process takes place both in the presence and absence of oxygen, which means that it is a common step in both aerobic and anaerobic respiration. The process takes place in cytosol. Glycolysis is actually a series of reactions, a pathway, and each reaction is catalyzed by a specific enzyme. All these enzymes are present in cytosol. Glycolysis needs glucose molecule, different enzymes, energy in the form of ATP molecules, and coenzymes, that is nicotinamide, adenine, dinucleotide in oxidized form. The end products of glycolysis are 2 pyruvates, 4 ATP molecules in total, and 2 ATP molecules are the net gain of glycolysis, and 2 coenzymes in the reduced form, that is NADH. Glycolysis can be divided into two phases. The first one is preparatory phase, which is also known as energy investment phase, in which 2 ATP molecules are utilized. The second phase is known as oxidative phase or energy production phase, during which 4 ATP molecules are produced. We will start with the preparatory phase. Preparatory phase is actually the phosphorylation and breaking of glucose molecule. Phosphorylation means addition of phosphate group. In the first reaction, glucose molecule is phosphorylated by 1 ATP molecule and is converted into glucose 6-phosphate. A high energy phosphate from ATP is transferred to the glucose molecule and the reaction is catalyzed by hexokinase enzyme. In the second reaction, glucose 6-phosphate is isomerized into fructose 6-phosphate. It is an isomerization reaction, and aldose sugar is converted into ketose sugar. The reaction is catalyzed by an enzyme known as phosphohexose kinase. Fructose 6-phosphate is again phosphorylated by a second ATP molecule into fructose 1,6-diphosphate. Another phosphate molecule from ATP is transferred to the fructose 6-phosphate molecule, which is converted into fructose 1,6-diphosphate. Two phosphate molecules are attached to the fructose molecule at carbon number 1 and carbon number 6. The reaction is catalyzed by phosphofructokinase. Fructose 1,6-diphosphate molecule splits into two 3-carbon molecules known as glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. The reaction is catalyzed by enzyme aldolase, and these two molecules, glyceraldehyde 3-phosphate and dihydroxyacetone phosphate, are isomers and are interconvertible. The equilibrium of the reaction is in the direction of glyceraldehyde 3-phosphate, because it is the glyceraldehyde 3-phosphate which is processed in the next step of glycolysis, that is known as oxidative phase. Dihydroxyacetone phosphate is converted into glyceraldehyde 3-phosphate by an enzyme known as triose phosphate isomerase. The glyceraldehyde 3-phosphate, from here on we will call it G3P molecule for convenience. In the oxidative phase we will count every molecule as two molecules, because dihydroxyacetone phosphate is converted into glyceraldehyde 3-phosphate, and from here on two molecules will be processed and every subsequent substrate or every subsequent molecule will be two copies. So in the oxidative phase, two hydrogens are transferred from glyceraldehyde 3-phosphate molecule to a coenzyme NAD, which is reduced into NADH. During this reaction, glyceraldehyde 3-phosphate is also oxidized into phosphoglyceric acid or PGA molecule. During this reaction, another phosphate molecule is also transferred to PGA molecule, so it is now a 1,3-PGA molecule. That is, two phosphate groups are attached at carbon number 1 and carbon number 3. This oxidation reaction is an energy yielding process, so a high energy phosphate bond is created in the 1,3-PGA molecule, that is represented here. This reaction is catalyzed by G3P dehydrogenase, that is glyceraldehyde 3-phosphate dehydrogenase. In the next reaction, 1,3-PGA or 1,3-diphosphoglyceric acid transfer its high energy phosphate molecule to ADP molecule, which is converted into ATP molecule. Remember that every reaction from here on will be considered to occur twice because two G3P molecules are produced at the end of preparatory phase. So two NAD molecules are reduced into two NADH and two molecules of 1,3-PGA molecules are produced. By transferring one of the phosphate molecule to ADP molecule, the ADP molecule is converted into ATP and 1,3-PGA molecule is converted into 3-phosphoglyceric acid or 3-PGA. This reaction is catalyzed by an enzyme known as phosphoglycerate kinase. In the next step, 3-phosphoglyceric acid is converted into 2-phosphoglyceric acid. It is an isomerization reaction in which only the position of phosphate group is changed. Here in this molecule, phosphate is attached to the carbon number 3 and in the 2-phosphoglyceric acid, phosphate molecule is attached to the carbon number 2. This reaction is catalyzed by an enzyme known as phosphoglycerate mutase. In the next step, a water molecule is removed from each of the phosphoglyceric acid molecule which is converted into phosphoenol pyruvate. The reaction is catalyzed by an enzyme known as enolase. As we have two molecules of phosphoglyceric acid here, so two molecules of water are produced at this step and two molecules of phosphoenol pyruvate are produced. In the last reaction of glycolysis, phosphoenol pyruvate transfer its high energy phosphate molecule to another ADP molecule which is converted into ATP. As we have two molecules of phosphoenol pyruvate, so two ATP molecules are produced at this step. This reaction is catalyzed by an enzyme known as pyruvate kinase. And phosphoenol pyruvate is converted into pyruvic acid in this final step. So at the end of glycolysis, we have two molecules of NADH, 4 ATP molecules, 2 produced in this reaction, and 2 molecules of ATP are produced in this reaction. And glucose molecule is converted or broken down into 2 molecules of pyruvic acid. A total of 4 ATP molecules are produced in the oxidative phase, but as we know that 2 molecules are already used in the first phase, the preparatory phase of glycolysis, so the net gain of energy in glycolysis is 2 ATP. The end product of glycolysis, that is pyruvic acid, it is further processed by pyruvate oxidation and Krebs cycle in the next steps of aerobic respiration. Pyruvate oxidation and Krebs cycle will be discussed in the next videos. This was all about glycolysis. Thank you for watching and please subscribe the channel for upcoming videos.